Okay, so welcome to video 4 in the series on G03, the read mapping file formats. So the previous one we talked about using linking read mapping with assembly, and in the next video we'll talk about the practical. So we're more focused on practical steps here and how we go from stage to stage to stage and how we get familiar with the file formats. So for overview, this is from an old article from 12 years ago by De Cristo et al., but I've outlines the stages. We're going from FASTQ here with the reads, and we're going to convert that into a map, a sequence alignment map containing our reads in the context of the reference genome to which we map them. Then we convert that into from a SAM to a BAM file. A BAM file is like a SAM file, except it's for the computer. You won't be able to look at it, whereas you will be able to look at a SAM file that is human readable. We'll convert the BAM in G04 into a BCF file, which is a binary variant call format. Again, it's a binary file, so you won't be able to look at it, but we will be able to look at it when you convert that from a BCF to a VCF. And you'll see similar summary elsewhere if you look at the Galaxy Training Network page, it kind of the same sort of stages, where this kind of BAM SAM processing is what we're going to also do in the alignment in the practical. But that will actually tell us a lot about the quality of our read mapping. So this is a summary here of what we're looking at. SAM is a sequence alignment map file. BAM is a binary SAM. BAI is actually a BAM index file. So it's a indexing file for efficient examination of that data. Then we do a VCF file, a genomic VCF file, which is a genome-wide VCF file. GFF is a general feature format, which is an annotation file, which is variations GFF, the original version, and GFF2 are slightly older. GFF3 is the current version. So a few pointers on this. So SAM files are very, very large, so you can delete them after you create them once you've converted them to the BAM format. You don't need to keep them, don't store them. They're very, very large. You can recreate them quite easily, and we'll do that. BAM files, we'll use them as they support fast random access. This access is permitted by indexing. So that means you can extract information very, very quickly, so that alarm reviewers don't decompress the band to look at the read info for particular coordinates. So hence we will have this file called the sample name x.bam.bai at the end. So it's a different file type, so just bear that in mind. The SAM file format we're going to have is something like this. So you can actually look at this when you have the files on your computer. It'll typically have a header of some series of lines. It might be shorter than the one shown here, because this is for a human example where there's a bunch of chromosomes. If we're dealing with a host or vector, maybe more like this. If we're dealing with a virus, it'll be more brief. So typically you have a header like this, and then a bunch of different chromosomes, and there you see the lengths there of the different chromosomes. And then we have information on the read library. So that might contain the platform library sample information that may or may not be present. And this, as you can read there, is an old example. And then further down is actual map reads here again, and that's contained over several lines, as you can see. And each contains an ID tag here, the sequence information, and then you have read quality information here. So we can look further at what's inside each read. So you have the ID, and if you have paired in reads, you want to match the partner together. It has, as per the name, it's a sequence alignment map. So you have the read position where that read maps, and then you have information on the alignment. So 37M indicates 37 matches, and this read is just 37 bases. So that's a pretty good match. And then you have the sequence here. So this is quite a short read. If we had a different code here instead of 37M, we'd have 13M, 17D, 24M, which would mean 13 matches, 17 deletions, and 24 matches. So that encoding inside there kind of tells you more about each read. In terms of the details, so we have the ID, the alignment flags here. reference name, so that may just be a single chromosome for a virus, the position, the mapping quality, which is high here, the CIGAR, compact idiosyncratic gapped alignment report is just simply the format. The ID of the mate, if it is paired end, so you see just an equal sign there. The position of the mate, which is base 282, and then the Fred scaled quality score across the whole read. So each of these reads is contained across 4 lines, just like the FASTQ file, and these are just the mapped, well, the SAM file will contain the mapped and unmapped reads, and they'll be denoted as unmapped at the coordinates to say if that is actually mapped or not. The VAM file is in the same format, same information, but it's just arranged as a kind of a single non-human readable format. So the read ID, the location where it's mapped, the alignment score, the sequence, the quality score, and other metadata associated with that. So it's just basically the same thing. So what we're going to do then is consider SAM and BAM files in the practical, and then we'll cover the other formats, BCF, BCF, GPCF, annotation files, and so on in G04. Okay, so we'll talk about the practical in the next video.